The overall goal of this procedure is to record the effect of the applied electric fields on neural progenitor cell migration in 2D culture and 3D ex vivo models. This is accomplished by first isolating and expanding murine neural progenitor cells in vitro. The second step is to prepare a single-cell suspension of neural progenitor cells. Next, for 2D culture preparation, neural progenitor cells are plated in custom-designed electrotactic chambers. Alternatively, for 3D ex vivo model preparation, neural progenitor cells are labelled with hooks 33342 and injected into a spinal cord slice organotypic model. Ultimately, an electric field is applied to the electrotactic chambers and time-lapse recording is performed to show that neural progenitor cells migrate towards the cathode of the electric field in both 2D and 3D environments.
The main advantage of this technique is that it provides a novel method for the live tracking of electric field-induced migration of individual cells following transplantation into a 3D organotypic model closely resembling the natural environment.
This method can not only provide insight into the electrotactic migration of neural progenitor cells transplanted into spinal cord slice, but can also be applied to the other systems, such as brain slice models.
To begin this procedure, prepare 22 by 11 mm strips of glass by dividing the autoclave 22 by 22 mm No. 1 thickness cover slips in half using a diamond pen. Next, create a freestanding glass well of interior dimensions 22 by